Hi class, let's finish up our discussion on diagnostic techniques by talking about some of the new kids on the block. These are the breakthrough or emerging methodologies that are increasing in prevalence but are still not very widely used. These methods are giving us a new golden age of diagnostic criteria or diagnostic techniques because they allow for very rapid, very quick, and very accurate collection and determination of incredibly large numbers of data sets. And these large data sets are increasingly going to require large computers to analyze and individuals with degrees in statistics to help you make sense of the results. Now, traditionally, we focused on isolating a microbe from pure culture and then reintroducing that microbe to an organism and then observing the same disease traits being in that new organism, and then identifying that microbe that's been pure cultured. So combining Koch's postulates with the traditional phenotypic and genotypic methods. But that takes a really long time to do. And what if you waste that time identifying a bystander bacterium, part of the normal microbiota? This traditional technique, this gold standard, takes a long time in that it's over 18 hours, and 18 hours when a patient is in critical condition is a big deal. Or what if this infection involves different microbial species working together within a biofilm? What then? So we need ways to work with large data sets and large microbial populations that are diverse. If you're working with a patient that has a bloodstream infection, aka septicemia, identifying the specific cause of septicemia can take a day. Now, this initial time frame for critical crisis management is typically going to be less than 6 hours, not 24 hours. So what is a physician, what is a healthcare provider going to do? Well, they're going to play it safe. They're typically going to give the patient a broad spectrum antibiotic and hope that they respond to it. If the patient does respond during that critical first hours, then hey, wonderful. They're going to keep receiving the same antibiotic because it's working well and the patient can recover. But if the patient does not improve, sometimes that broad spectrum antibiotic that was given to them initially was the inappropriate prescription. And as a result of receiving the inappropriate treatment during that critical time period, those first 6 hours, the patient may or may not recover all the way. They may accidentally die. So we need a more sophisticated diagnostic technique that can get us the correct diagnosis in a very short period of time, ideally less than that 6 hour period. So I want to introduce you to microarrays. I love microarrays. I worked with microarrays when I was doing my undergraduate research projects for genome-wide analysis of Arabidopsis and Brassica species. These are essentially DNA computer chips. They're super duper cool. And it boggles my mind that we're capable of making these. And when I say we, I mean the human species as a whole. What we'll do is we'll take a computer chip. So we'll take a chunk of silicone and we'll break it up into sections. And I'm grossly simplifying this right now. And in each section, we'll have different DNA fragments. And those different DNA fragments are going to be single-stranded DNA fragments with specific genetic codes to them. So each DNA fragment that we put on our segment of the microarray has a specific genetic code. We'll then take a genome-wide sample from our organism, expose it to some endonucleases to chop up its genome into little pieces, and then those little pieces of the microbe's genome will stick to different regions of our microarray chip that have the specific DNA fragments on it. And we can determine what our organism is based on which segments on the plate, which wells on our microarray plate have hybridization occurring. Now, these wells on the microarray are itty bitty. They're very tiny. And we typically are going to need a computer to help us analyze these incredibly large data sets. So that chip will be ran through a computer system, exposed to fluorescent light, and then that data will be processed via some stats programs. We also have nucleic acid sequencing. Sometimes we could just sequence the entire genome of the microbe on the spot to determine what the diagnosis is of that microbe. The nice side effect of the human genome project, is that now genome analysis is much lower in cost, and it's much quicker, and it's much more ubiquitous. So it's now starting to become commonplace in technical research institutions. Most major research universities now have nucleic acid sequencing technology, and it might start to become commonplace in larger hospitals or clinics. This next generation of sequencing is going to focus on random primers being linked throughout the entire genome, which will give us novel nucleic acid sequences, or identification of our novel nucleic acid sequences. Recall, a primer is a small segment of DNA or RNA, a small segment of nucleic acids that's used to initiate the synthesis of a new nucleic acid. So when we're synthesizing new DNA, we'll start with single-stranded DNA, attach the primer, and then DNA polymerase can attach to the single strand and start synthesizing or replicating the genome. This deep sequencing of the entire genome with the random primers can be analyzed multiple times and scanned very rapidly to minimize the amount of errors. Potentially in the future, we could take a sample from the patient and sequence all of the microbes in it very quickly, very rapidly, and then determine a complete profile of the exact microbes that are present, so we can have a much more accurate diagnosis and know exactly at a genomic level which medications, which antibiotics those microbes are resistant to. Another technique is mass spectrometry. In a nutshell, mass spectrometry involves taking the sample and breaking up the samples into itty-bitty tiny molecules and fragments, ionizing those fragments, and using an electrical current to throw them into an electromagnetic plate. And based on where they hit the electromagnetic plate, we can determine the exact chemical composition of those fragments. Now, we can use this technique with larger biological molecules with a little bit of modification. Generally speaking, this is used with smaller inorganic molecules, and they're very heavily ionized and aggressively broken up. But with incredibly large biological molecules, that's aggressive ionization and fragmentation is not advantageous. We still want to keep the protein molecule more or less intact. We just want it to be fragmented from its associated protein molecules, so it's no longer hybridized to them, and we also want it to be ionized at the same time. So the twist, or the new form of mass spectrometry that's being used is called a MALDI-TOF mass spec. MALDI-TOF is kind of a mouthful of an acronym. It stands for Matrix-Assisted Laser Deionization Time of Flight Mass Spectrometry. In other words, we measure how big the protein samples are, and by measuring the exact mass of the protein sample, we can determine the exact chemical composition of the protein sample. And by measuring what proteins are present, we can thereby deduce what bacterial, viral, or fungal species are present in our sample. We've also are now employing medical imaging to help us make more accurate diagnosis and treatments of diseases. MRIs, CT scans, and PET scans are increasingly being employed to find the location of infection in deep tissues that are normally not accessible without exploratory surgery. For centuries now, since the 1800s, we've been using X-rays to help diagnose the presence of a tuberculosis infection in the lungs. A chest X-ray to determine tuberculosis of the lungs is one of the standard techniques that's still employed today. This is a lovely figure that is showing us, first, on this side, how a microarray works. So here we can see that there are many, many, many different little well plates, and those itty-bitty little well plates are going to be exposed to different wavelengths of lasers that cause them to fluoresce different colors depending on what kind of hybridization occurs. And then the computer analyzes this sample. Up here, we can see the results from our random primers sequencing the entire genome of the microorganism. And we can find areas of overlap to help us piece together the puzzle of the entire microorganism's genome. And then finally, in the bottom, we have some data from a MALDI-TOF mass spec. And then right here, we can see the results of an MRI scan that's been used to peer inside of a patient to let us know what is going on in their body, what structures are present, and ideally where the infections are located. So, concept check class. Which of the following breakthrough methodologies utilizes absorbent plates containing gene sequences from possible infections? So which technique involves a computer chip that has DNA or gene sequences attached to it? Is it a nucleic acid sequencing, mass spec, microarrays, imaging techniques, or MALDI-TOF? Pause the video, rewind it, check your notes, get me an answer. 1, 2, 3, 4, 5. The correct answer is C, microarrays. Microarrays are dependent upon many known samples of DNA being bound to a computer chip and testing whether or not they hybridize with an unknown sample. If you have any questions about this material, please feel free to post them in the discussion board or get back to me or with an email. As always, happy studying.